Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is the animal science. My dear students, in the last lecture, we have learned about the process of formation of RBC, which is known as what? Erythropoiesis. It comes under the process of formation of different blood cellular components like WBCs, platelets, and the lymphocytes as well. The process was hematopoiesis. We discussed that how hematopoiesis in the stem cells, which are pluripotent, which can be converted into any form, they are converted into myeloid stem cell and lymphoid stem cell. The myeloid stem cell, they basically are responsible for the precursors of WBCs, RBCs, and the other cells. And the lymphoid stem cells, they form the B lymphocytes, T lymphocytes, and the natural killer cells. So now we are going to discuss about the different types of leukocytes, means the white blood corpuscles. Okay. Before starting with this topic, there are two terms: DLC, which is differential leukocyte count, differential leukocyte count, and TLC, that is total leukocyte count. So first is the differential leukocyte count, that is DLC, and the second is the total leukocyte count, that is TLC. So the difference between different types of leukocytes, okay. Now we know that eosinophils, basophils, and neutrophils, they belong to the category of granulocytes. Okay, eosinophils, basophils, and neutrophils, they basically belong to the category of granulocytes, and monocytes and lymphocytes, they belong to the category of agranulocytes. Agranulocytes simply means what? That they are not having granules, and granulocytes means what? That granules are present. So in agranulocytes, lymphocyte, it is dominantly present, and in case of granulocytes, the neutrophils are dominantly present. Understood? So the total leukocyte count and the differential leukocyte count basically says what? The total leukocyte count, or TLC, it gives a picture that around 4,000 to 11,000 cells are present per cubic mm. When TLC says what? That per millimeter cube of blood, around 4,000 to 11,000 cells are present. Out of these cells, how many are neutrophils, how many are basophils, how many are eosinophils, how many are lymphocytes, and how many monocytes? So this is what the differential leukocyte count. Understood? So lymphocytes around 30%. Okay. Monocytes around 5.3%. Eosinophils around 2.3%. Basophils around 0.4%, and neutrophils around 62%. Understood? So, granules in the cytoplasm. In lymphocytes, the granules are absent. Monocytes, the granules are absent. Eosinophil granules are present and they are coarse. Okay? Mean rough. Basophils, coarse. Neutrophils, the granules are fine. Understood? So the granules present in the cytoplasm in case of eosinophils are coarse, basophils also same, and neutrophils, they are fine. Is it clear? Yeah. Now coming to next, staining of cytoplasm. Eosinophils, they are eosinophilic, mean acidic stain. Basophils, basophilic mean basic stain, and neutrophils, neutrophilic means towards neutral. Okay, so neutrophils, they basically shows neutrophilic nature, means they are stained by both acidic and basic stains. So both acidic and basic acidic stain. Understood? Now coming to next, nucleus in case of lymphocytes is rounded. In monocytes it is bean shaped. Okay. In eosinophils it is bilobed. In basophils it is S-shaped or three-lobed, and in neutrophils it is multi-lobed, 3 to 7. So nucleus in case of lymphocytes is rounded. In case of monocytes it is bean-shaped. In case of eosinophils it is bilobed. In case of neutrophils it is multi-lobed, and in case of basophils it is S-shaped or three-lobed. Site of formation. In lymphocytes, site of formation is lymph nodes, spleen, thymus, tonsils, Peyer's patches, bone marrow. Understood? In lymphocytes, the site of formation of lymphocytes are lymph nodes, spleen, thymus, Peyer's patches, bone marrow, and tonsils. Monocytes, the site of formation is bone marrow. Okay. Eosinophils, the site of formation is bone marrow. Basophils, bone marrow. Neutrophils, bone marrow. So all the other leukocytes, their site of formation is bone marrow. Lymphocytes, they form in the lymph nodes, spleen, thymus, Peyer's patches, and bone marrow. Lifespan, lymphocytes few days or even years. Monocytes 10 to 20 hours in the blood tissue, month or even years. Eosinophils 4 to 8 hours in blood or 4 to 5 days in tissue. Basophil 4 to 8 hours in blood and 4 to 5 days in tissue. Same in case for neutrophils. The eosinophils, basophils, and neutrophils, their lifespan in blood, freely flowing blood, is 4 to 8 hours. While in case of monocytes, the lifespan is 10 to 20 hours in the blood. Okay. And in lymphocytes, same, fewer days. Functional lymphocytes, it is concerned with antibody formation. Monocytes, phagocytic. Eosinophils, important role in immunity or anti-allergic. Basophils, they help in the secretion of heparin, histamine, and serotonin, mean all the cell mediators. And neutrophils, phagocytic. So neutrophils and monocytes, they are phagocytic. Okay. So lymphocytes, they are concerned with antibody formation. Monocytes, they are phagocytic. Eosinophils, they have important role in immunity or anti-allergic. Basophils, they help in the secretion of heparin, histamine, and serotonin. And neutrophils, they are also phagocytic in nature. Understood? Types of leukocytes. Differential leukocytes, there are two types: granulocytes with granules in the cytoplasm and agranulocytes without granules in the cytoplasm. Granules, three types on the basis of staining characteristics of cytoplasm, and granules in the shape of nucleus. Okay. There are further of three types: neutrophils, eosinophils, and basophils. 62%, eosinophils 2.3%, and basophils 0.4%. Agranulocytes without granules, monocytes 5.3%, and lymphocytes 30%. Okay. So agranulocytes, they are further divided into monocytes and agranulocytes, lymphocytes. Monocytes 5.3% and lymphocytes around 30%. Okay. Leukocytes. White blood corpuscles are also called the leukocytes because they are colorless. TLC, total leukocyte count, already told you it is 4,000 to 11,000, not 8,000. It is 4,000 to 11,000. Leukocytosis, increase in TLC. This condition is in bacterial or viral infection. Leukocytosis is the increase in the amount of WBCs, or the total leukocyte count. Leukocytopenia, decrease in total leukocyte count. Normally TLC increases in bacterial and viral infection, but in typhoid and AIDS, TLC decreases. Leukemia, abnormal increase in TLC more than 1 lakh, it is called as blood cancer. Leukemia is an abnormal increase in total leukocyte count more than 1 lakh, it is called as blood cancer. On the basis of nucleus and nature of cytoplasm, leukocytes are of two types. Granulocytes, in their cytoplasm, granules may be present which can be stained by specific dye. Nucleus may be multi-lobed and lobes are interconnected by protoplasmic strands produced in bone marrow. So granulocytes, they all produce in bone marrow. Okay, they have cytoplasmic granules. These cytoplasmic granules, they can be the modification of rRNA, ribosomal RNA particles. They are known as chromaffin granules because they are lovable or having affinity towards color or dye. Understood? So these granules are basically rRNA particles which are chromaffin granules mean affinited towards a particular dye. Nucleus may be multi-lobed, okay, and lobes are interconnected by protoplasmic strand produced in bone marrow. They are acidophils, basophils, and neutrophils. Acidophils, they are also known as the eosinophils, okay. Agranulocyte, cytoplasm is clear and granular, clear and non-granular, agranular. Nucleus do not divide in lobes, so called mononuclear WBC, okay. When nucleus is not divided in lobes, that's why it is known as what mononuclear WBC. Produced in bone marrow, they are of two types, monocytes and lymphocytes. Monocytes and lymphocytes. Understood? Now, granulocytes. Acidophils, also known as eosinophils, they are amoeboid in shape. Nucleus is having two lobes. Nucleus is two lobed, okay. They have coarse granules. Their granules take acidic stain, eosin. So they are basically stained by a acidic stain. And that acidic stain is known as what? Eosin. Acidic stain. Understood? Their number increases in people with allergic conditions such as asthma or hay fever. Increase in number of acidophils condition means allergic conditions like eosinophilia occurs in teniasis, ascariasis, hay fever, parasitic infection, and asthma. Okay. They also help in dissolving blood clot. They are non-phagocytic. Non-phagocytic. Understood? Eosinophils or acidophils, they are somewhat similar to lysosomes. Eosinophils can attach to the parasitic forms and cause their destruction by increasing the concentration of lysozymal enzymes like acid phosphatases and acid and other enzymes. Okay, so they look like lysozymes, lysozymal function. They attach to parasite and destruct it. Understood? Now, basophils. Basophils, they usually have three lobes. Usually. Minimum in number, amoeboid in shape, size 8 to 10 micrometers. Okay. Smallest granulocytes. They are smallest granulocytes. Okay. And their main function is to secrete the cell mediators like histamine, serotonin, and heparin. Okay. They are basically stained by basic stain. Understood? Both mast cells and basophils liberate histamine and heparin as well as smaller quantities of other cell mediators. Both mast cells and basophils secrete heparin and histamine. Understood? They also help to secrete bradykinin and serotonin in small amount. Okay, already mentioned. So basophils can be considered as mast cells of connective tissue. They are known as mast cells of connective tissue. Understood? Now, coming to next, neutrophils or heterophils. Neutrophils also known as heterophils. They are maximum in number, amoeboid in shape, size 10 to 12 micrometers. The lobes present in the nucleus, okay, they are multi-lobed. Counting the lobes of neutrophils is called Arnett count. It is known as what? Arnett count means the number of lobes present. They are active, motile WBC. So neutrophils or heterophils, they are active and motile WBC. They can squeeze and comes out from the wall of blood capillaries. Okay, this phenomenon is called diapedesis. So neutrophils or heterophils, they can squeeze and come out of from the blood capillaries in the tissue, and this is known as what? Diapedesis. They are phagocytic in nature. Neutrophils, they are phagocytic in nature. Okay, they destroy bacteria and virus by phagocytosis. Due to their smaller size and phagocytic nature, they are called micropolicemen, known as micropolicemen. Okay, neutrophils. The counting of the lobes of neutrophils is known as what? Arnett count. They are active and motile WBC. They can squeeze and comes out of the blood capillaries in the tissue. This phenomenon is called diapedesis. They are phagocytic in nature. They destroy bacteria and virus by phagocytosis. Due to their smaller size and phagocytic nature, they are called micropolicemen. They are called micropolicemen. They help in detection of sex. They help in detection of sex. In female neutrophils, bar body is attached. Okay. In female neutrophils, bar body is attached with the lobe of nucleus, which is formed by the modification of X chromosomes. Bar body is absent in males. So these bar bodies which are present in females, they can be a part of, justification or, diagnosis of sexual determination. Okay, so they help in sex detection. That is why it is written, okay, that the females neutrophils, they have bar body, but the males, they don't have bar body. Understood? Bar body means one extra chromosome. Like for example, females, they have, you know like this. So one is considered as X chromosome and the other one is considered as a bar body. Males have XY, so they have only one X chromosome, no bar body. Understood? Neutrophils, they are also known as what? Heterophils. Okay, nucleus is multi-lobed. Most importantly, one point, that in case of neutrophils, the multi-lobed, this multi-lobed appearance is known as what? Drumstick. Okay, or it is basically known as bar body. So this bar body is also known as or called drumstick. Okay, drumstick. Understood? Then agranulocytes. Agranulocytes mean they don't have granules. Example, monocytes and lymphocytes. Understood? So monocytes, they are the largest of all types of leukocytes. Largest. Monocytes are the largest blood corpuscles. Size 12 to 20 microns. Power of diapedesis is present just like neutrophils. Active motile WBC. They have active motile WBC when they are also concerned with phagocytosis. Destroy bacteria and virus by the phagocytosis, so-called macropolicemen. They are known as what? Macropolicemen. So monocytes, they are known as what? Macropolicemen, and neutrophils, micropolicemen. Understood? Also called scavenger of blood because they engulf damaged or dead and minute bits of blood corpuscles. Whatever is which is not of use, waste, it will be digested by these monocytes. Lymphocytes, they are amoeboid in shape, size 6 to 16 microns, smallest WBC. Lymphocytes, they are the smallest WBC, and lymphocytes are of two types. So lymphocytes, they are the smallest WBCs. Okay, they are non-motile, they are non-phagocytic. Okay, they are the smallest WBCs. Understood? They mainly helps to form, and it is of two types: B lymphocytes and T lymphocytes. Okay, lymphocytes. There are two types of lymphocytes: B lymphocytes and T lymphocytes. T lymphocytes. T lymphocytes, they are responsible for cell-mediated immunity. They are produced in bone marrow but mature in thymus gland. That is why they are given the name T lymphocyte, because of this thymus. So T lymphocytes, they are produced in bone marrow and they are matured in thymus. That is why they are known as cell-mediated immunity or the T lymphocytes. On the basis of function of T lymphocytes, it is of four types: T killer or cytotoxic T cells. They are denoted by like this. T helper, which stimulate the B lymphocytes to produce antibodies, TH. T suppressor, which suppresses the T killer cells and protect immune system. T suppressors, they suppress the T killer cells to protect the immune system. They maintain the balance. Killer lymphocytes. Right? Then the memory T cells. These memory T cells, they basically, they have a memory which remember the pathogen and the response developed by body. Okay. So these T memory cells, they basically help to store the profile of bacteria or virus or a protein. Okay. So that anaphylactic The secondary response when for the first time the pathogen enters the body. The memory T cells, they will remember the attack of that pathogen, and now the second time when that pathogen tries to enter, the antibodies will be initiated or it will be generated in a very frequent rate. Okay, this is known as what, or this is the function of memory T cells. Understood? Yeah. B lymphocytes, they are responsible for humoral immunity. Okay, so T lymphocytes and B lymphocytes. B lymphocytes, the word B is basically taken from Bursa fabricus. In case of birds, the B lymphocytes, they are produced in bone marrow and they are mature in bone marrow. And the bone marrow in case of, you know, birds is known as what? Bursa fabricus. Its function is to produce, as birds they have pneumatic bones without marrow. Its function is to produce and synthesize and transport antibodies. Okay. So B lymphocytes, they produce in bone marrow and they mature in bone marrow, and their main function is to produce and synthesize antibodies. Understood? Now, this is the stem cell pathway. This is the hematopoietic stem cell or hemocytoblast, which differentiate to form myeloid stem cell and lymphoid stem cell. Myeloid stem cell, it further forms myeloblast, 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 and monoblast. Okay. These myeloblasts, they further form promyelocytes, okay, which further forms eosinophils and basophils myelocytes, which further form eosinophils and basophils respectively. Understood? The myeloblast, it forms the neutrophil myelocyte, okay, which forms the neutrophils. Monoblast, it forms promonocyte, then monocyte, and monocytes, when they enter the tissue, they form macrophages. Understood? So monocytes macrophage system. Is it clear? Now, coming to next, lymphoid stem cells, they form B lymphocyte, T lymphocyte precursors. They form B lymphocytes and T lymphocytes respectively. Okay. B lymphocytes, they also differentiate to form plasma cells and memory cells. And T lymphocytes, they form effector T cells, helper T cells, and like that. Okay. So this is stem cell pathway which help us to, now T cells, they don't release antibodies, but they attack the antigens. They attack antigens. And that is why they also attack the transplants and the cancerous cells. So T cells, they attack the graft or the transplants and the cancerous cells. Okay. Understood? Yeah. Platelets. Platelets, they are also known as thrombocytes. Okay. Also known as thrombocytes. Found only in mammals, while in other vertebrates, spindle corpuscles are present which perform same function. So platelets or thrombocytes, they are mainly found in mammals, okay, while in other vertebrates, spindle corpuscles are present which perform same function. Understood? They are non-nucleated and derived from megakaryocytes, cells of bone marrow. They are non-nucleated and they are derived from megakaryocytes, very large cells of the bone marrow. Okay. Platelets are disc-like, oval-shaped or biconvex. These are the probable shapes: disc-like, oval-shaped or biconvex. While spindle corpuscles are spindle in shape and round nucleus is present in the center. In their cytoplasm, basophilic granules are present which can be stained by methylene blue. So platelets, they can be stained by methylene blue. Okay, because it is a basophilic granule. Maximum part of cytoplasm is composed of contractile protein thrombostenin. The thrombostenin is a contractile protein which is present in the cytoplasm of platelets. Understood? Size 2 to 3 micrometers, lifespan 2 to 4 or 5 days. Count 1.5 to 4.5 lakhs per cubic mm, normal count. Decrease in number of blood platelets is known as what? Thrombocytopenia. And increase in number, thrombocytosis. Okay. Critical amount of thrombocytes is 40,000. It should not be below this level. If number less than this level, then it becomes thrombocytopenic purpura. The disease is thrombocytopenic purpura. In this disease, because of very low amount of platelets, any a very small trauma or a pressure can create the blood clotting. Okay. Red spot or rashes appears on the skin. Okay, called purpura disease. So because of this, we can say less amount of thrombocytes or the platelets in the blood, the bleeding can happen and it can be very versatile even the trauma is very less or it is not so severe. They help to repair the endothelium, which is the wall of blood vessels, blood vascular system, by the formation of platelet plug, because they have tendency to attach on the gelatinous or mucilaginous surface. They have tendency to attach on the gelatin or the mucin or mucus containing surface. Synthesize thromboplastin, which help in blood clotting. Platelets, they help to synthesize thromboplastin, which helps in blood clotting, and they also synthesize serotonin. So these are the functions of platelets. These are, you can see the activated platelets are seen. Okay. Now we will be considering or we will be doing the hemopoiesis is already done. Okay. I will be starting the part of the lymph in the next class. Okay. And before going to the lymph part, we will be discussing the blood clotting factors, the blood clotting, the cascade of clotting. Okay. Till then, my dear students, till my next class, please see the previous lectures, videos, so that you could adapt a better connect for the coming videos. Till then, bye bye, take care, stay safe. Bye.